So for those of you who do not know me, I'm Barbara Tate. I'm the Chief Scientific Officer at FARA, and I have the privilege today to moderate a panel of our industry partners who have the most advanced programs in the pipeline. These are programs that have actually entered human clinical trials. But before that, I wanted to just give you an overview of FARA's vision for the pipeline. As you heard from the previous presentation, FA is very complex. And that complexity means that there isn't going to be a single treatment that helps everybody at every stage of FA with all their symptoms. So what we need, and I think you've seen this before, is a shots on goal approach. We need multiple effective therapeutic approaches that together can treat everyone with FA. And we are excited that we have our first goal, Skyclarys, approved in the US. And you'll hear from the Skyclarys team and from the Biogen team in just a few minutes. But in addition to that, we have other treatments in the pipeline that will miss the goal. That's disappointing, but it's not a failure if we learn. We need to learn from those that miss the goal as well as those that hit the goal. And ultimately, we're looking for a variety of complementary mechanisms that together give us the holistic treatment for FA. We often call this a cocktail approach. That is that there's layers of treatments that together allow us to address all the symptoms of the disease. How do we get to a cocktail approach? Well, it's from you and your friends and family's support of FARA that has led us to develop the basic understanding of FA. It is on that foundation of the basic science that we can find treatments. As you've heard, and I think you all know, FA is a genetic disease, a change in the DNA within the gene for the protein frataxin. Everybody has a frataxin gene, in fact, two, one from mom and one from dad. And everybody has within that frataxin gene a repeat of 3 of the DNA letters, GAA. Everybody has that. Most people have the GAA repeat in single digits, GAA, up to say 6. For some reasons that we do not understand, those letters expand. So that's the GAA repeat expansion that you've heard. And some people then have GAA in the numbers of tens or hundreds or even 1,000 or above 1,000. When the GAA repeats expand, the gene that produces frataxin cannot effectively make the protein. It makes some, but there's a deficit, less protein than we need to not have symptoms. And you heard from Dave earlier, carriers who have one expanded gene are asymptomatic. So we don't need to get to 100% of frataxin protein, but we need to get to at least 50% of what's expressed in people without expansions. And Dave also referred to the mitochondria. This is an organelle within a cell that's critical to cellular function. Frataxin has a role within the mitochondria. And when the mitochondria function is impaired, that will eventually lead to the symptomology of FA. And in 2 ways. There's the lack of function of the mitochondria, but also a buildup of byproducts of biochemical reactions. And the buildup is not healthy for the cell. And the cell looks very unhealthy. So we often refer to the energy of the cell, but there are other aspects of cellular function that are impacted in FA. And ultimately, this leads to the changes at the level of the organ, the brain, the heart, the spinal cord, that we know as FA. I want to return to the mitochondria for just a moment. You've all heard about it as the powerhouse of the cell, the site where energy is produced for the body. But as I referred to earlier, there are other biochemical reactions that take place in the mitochondria. And frataxin appears to be critical for those biochemical reactions to take place normally. When we do not have frataxin, we have low energy. But as I said, we also have a buildup of things like you've heard of reactive oxygen species, iron. And then there are mechanisms that are repair mechanisms for every cell that are also influenced by the lack of frataxin. So we don't repair damage to the cells as easily without frataxin. So just to some of these words that you've heard about: oxidative stress, iron sulfur cluster deficiency, iron deposits. These are all things that can occur in the absence of frataxin. What we're looking for is a cocktail, again, of treatments that either address, as Dave talked about with Riata, at the level downstream of loss of frataxin, so preventing or slowing some of those consequences of loss of low levels of frataxin, or can supplement the functions for which frataxin is essential, or even bypass the need for frataxin, an area of research now, or we can increase the levels of frataxin by different mechanisms. Together, and it is together that we need these complementary mechanisms in order to effectively address FA. The treatment targets come from this fundamental understanding of the disease. And you heard the panel before talk about symptomology. So scoliosis surgery, to be able to treat some of the symptoms, although we're not treating FA particularly, or heart medication to treat the heart. That is at the organ level. We're looking for things that can help cellular function. So even in the absence of functional mitochondria, can we keep the cells healthier for a longer period of time? You'll hear a lot today about mitochondrial approaches. That's been an area of focus in the pipeline. And as we heard earlier, that's where Riata's Skyclarys has its impact. Beyond that, there are things in the pipeline now that you're going to hear about today, which address the low levels of frataxin, either actually giving the frataxin protein or reactivating that silenced, frataxin gene to produce more frataxin. And finally, a genetic approach, giving people an actual gene for frataxin that does not have the pathological expansions of GAA. You've seen the treatment pipeline, and we color-coded this, so it all kind of comes together. Thank you, Maureen Yipe, for these beautiful slides. So the pipeline in this color coding tells you, most advanced at the top, which is Skyclarys, but also noted that a lot of the approaches, again, were focused on mitochondria. Beyond that, we're looking to go further upstream to try to address the loss of frataxin and even to replace the frataxin gene. So today we have a number of companies with us, which again, we thank the panelists for making the trip and joining us today. The community really does love to hear from you and to understand your programs. And each panelist will give us a few minutes talking about their specific programs, and then we will take questions. We're going to start with Riata, a Biogen company, and a Biogen colleague. Oh, did I do that? We'll be moving on to PTC, who unfortunately could not join us today. So those slides will be me presenting them. We'll hear from Stealth, as you see on our pipeline as well. The Larimar team, representative from the Larimar team, from the design team, and finally from Lexeo. So thank you all for everything you've done to contribute to get the pipeline where it is today. And I think we will start with our Reata columns. What's up?
Well, hi, everyone. It's nice to see so many familiar faces. And if I've not had the opportunity to meet you, please do come say hi to myself or any of the Biogen colleagues that are here with us today. For those of you that I've not had the opportunity to meet, my name is Payal. I did work with Reata and am now with Biogen. So very excited to be here. Thank you to FARA. This is a very unique time in Reata, now Biogen's life cycle, where we've gone through quite the development journey with omaveloxolone, otherwise known as Skyclarys, and I'm excited to be here to share some information with you. Before I get started, I do have to make a few disclaimers. First and foremost, the presentation on clinical trial data and Skyclarys or omaveloxolone is for informational purposes only. OMAV is indicated or has been approved by the FDA for the treatment of FA in patients 16 years and older. Please speak to your physician to determine if OMAV is the appropriate treatment option for you. So bear with me. I'm going to try and get through these slides as quickly as possible given our time limit, but I do have to compliantly share some of the important safety information for OMAV. Dr. Lynch did a nice job, but I do have to reiterate some of the fine points here. So of course, OMAV is used for the treatment of FA in adults and children 16 years of age and older. It is not known if OMAV is safe and effective for use in children younger than 16 at this time. Dr. Lynch did speak to some of the common side effects, excuse me. So I won't go into many of the details, but do know that OMAV does potentially increase transaminases. So if you are eligible for drug, know that your doctor will be monitoring blood levels of these transaminases prior to starting, during initiation of drug, and at periodic times after you start treatment. And don't be alarmed if there's some changes in doses that your doctor has to make based on these transaminase levels. So quickly, Jen did a very nice job of discussing what it takes to get a drug development process to the finish line. When a company sets out to develop a novel therapy in a rare disease space that has few or no treatments at all, it truly does take a village. And this is very much the case with omaveloxolone. First and foremost, the collaboration with FARA. They were critical back in 2014 or 2015, and absolutely critical in discussing the development program of omaveloxolone. The FDA, they recognized the need for a treatment in a rare disease space like FA. So their constant guidance, their discussions regarding analyzing the data was absolutely pertinent in the approval of omaveloxolone. And last but not least, all of you, for the community that participated in clinical trials, that are participating in the natural history study, that raised the voice in the petition to come back to FDA and Reata and have them revisit omaveloxolone in FA was absolutely critical in getting this to the finish line. So huge kudos to all of you in this room. To have an approval on Rare Disease Day also highlights just the awareness that's been put in a rare disease like FA. So, to receive this approval back in February was a huge milestone all the way around. And most recently, you may be aware that Reata was acquired by Biogen. And I think with that comes a lot of growth. And what I do know is that the two companies this was just announced 2 weeks ago. So we're right on the heels of this announcement. And what I do know so far is that there is this commitment to ensuring integration so that you experience continuity of care continuity of medication, and comes with this growth. And we'll talk a little bit about that. And I know my colleague Wendy is here, who on behalf of Biogen will share some highlights with Biogen's commitment to the rare disease space. So omavaloxolone was studied in a clinical trial, and that was called MOXIE. MOXIE, I like to think, was separated into 3 different categories. Part 1 was the dose ranging study. Part 2, which we'll talk about here, and then part 3 is the extension study. Due to limited time, we won't go through all components of it, but for the part 2 study, it was a phase 2 international double-blind randomized placebo-controlled parallel group trial, lots of words, I know. To be eligible for this trial, you had to be between the ages of 16 and 40 with a confirmed diagnosis of FA. Your mFARS score, now I'll take a quick pause. mFARS is a neurological assessment to determine how your FA is progressing. And you may notice your neurologist doing an mFARS scale assessment in the office. It measures 4 categories: upright stability, lower limb stability, upper limb stability, and mouth and throat function. Did I get all of them? So the goal was to determine at the end of 48 weeks, was there a difference in this mFARS score? So at the beginning of the trial, there was 103 patients enrolled in the MOXIE Part 2. They were separated into 2 groups. One group received omavaloxolone 150 milligrams. The other group received placebo. So again, the goal was to determine at the end of 48 weeks, was there a difference in the mFARS score between the group in the omavaloxolone treated group and in the placebo versus the placebo group. I'll have to take another pause here to give you some important safety information. Omavaloxolone also potentially may increase your BNP. You heard the physicians prior to talk about BNP. Again, this is just a measure of how well your heart is working. So your doctor will continue to check your BNP levels while you are on omavaloxolone. It's important to communicate with them if you notice any signs of fluid overload, such as sudden weight gain or swelling in your arms and legs, as well as the doctors mentioned some changes in your cholesterol levels. You'll notice your good cholesterol, which is your HDL, or your bad cholesterol, HDL, may go through some level changes. So know that your doctors will also be monitoring that. So this slide talks about the efficacy of omavaloxolone at the end of 48 weeks. When you look at the x-axis, which is your horizontal axis, you see the study week from 0 to 48. On the y-axis, you notice your change in mFARS score. 0, which is right in the middle there, is your baseline. I'll also highlight placebo is noted in that gray line, and then OMAV treated group is highlighted in the blue line. And what we're seeing at the end of 48 weeks is the line goes down for omavaloxolone. And in this case, that's a good thing. That means there's less physical impairment in the OMAV treated group at the end of 48 weeks as compared to placebo. Yet another safety information slide. So, and I'll talk about the most common side effects in a moment here, but we've been talking about increased liver enzymes, which is otherwise known as AST and ALT, headache, nausea, stomach pain, diarrhea. And then, as always, you want to make sure when discussing if Skyclarys is an appropriate treatment option for you, is to discuss any medical conditions with your physician prior to getting started. As I noted, these are the safety data that was collected in the 48-week Part 2 study. This is not all comprehensive, and I'll just highlight the top few. Again, elevated liver enzymes, headache, nausea, abdominal pain, fatigue, and diarrhea. And you'll see that they're separated into whether the group was in the placebo arm or whether they were in the OMAV treated arm. as Dr. Lynch was mentioning, it's very important to communicate all prescription medications and non-prescription supplements or medications that you may be taking with your physician. And what should I avoid while taking Skyclarys? Grapefruit juice, grapefruit fruit. Just don't take grapefruit. As always, as I mentioned, this is not an all comprehensive list of side effects. We're learning a lot more about OMAV the longer folks are on it. So if you notice any side effects, please do communicate that to your physician. Finally, I'll end with, OMAV has gone through quite the journey. And what you're seeing on this slide is only a snapshot of what's happened in the last 4 to 5 years. What I can say is, given the recent acquisition of Biogen, know that your questions and your want for information is certainly being heard. We are aware of the unmet need, especially when it comes to the pediatric trial and global access to drug. So know that we are still in the works. We're still trying to develop plans, get details. And so Biogen is committed to ensuring equitable access to drug moving forward. And this will come in the form of continued data generation, a real-world evidence study to better understand the safety and efficacy of omaveloxolone. This will also include future clinical trials. So know that there is this commitment to continue the momentum with omaveloxolone in the FA space. And I'll end with saying thank you to all of you for your participation and your involvement to get us to where we are today. So thank you.
And next we have someone from Biogen. Wendy, do you want to introduce yourself?
Happy to. Thank you so much. Yes, I'll wait for slides to come up. First of all, a huge thank you to FARA and to CHOP and just giving us the opportunity to be here with you today. This is as Biogen first FARA event, and we couldn't be more thrilled to be here with you. And thank you for embracing us. And just in the evening and into today, I've really enjoyed the conversations and learning from you all. So with that, we'll go ahead and get started with just an introduction to who we are as Biogen. So we are one of the first biotech companies that was started in 1978 by 2 Nobel Prize winners. And since then, we've really, with that rooted, grounded expertise in neuroscience, have remained committed to the space, really focused on the discovery as well as the development and then delivering therapies in the neuroscience rare disease space to communities with high unmet need. So given that we have this shared learning across neuromuscular rare disease, it gives us the opportunity to really have a clear understanding of the pathophysiology, the biology with the diseases that we study. And then what we can do is with the portfolio of our pipeline assets and what we bring to commercialization, we can look for those expertise, the shared learnings. What's that red thread between all of our different research programs that we can then bring forward those transferable learnings to bring more therapies to communities that need them. So with that, as Payal mentioned, we did announce the acquisition of Reata. So that closed on September 26th, so just 2 short weeks ago. And with this, it really, again, brings the opportunity to integrate all of the experiences from the Reata team, everything they've accomplished with you all in the community, with research programs, with understanding the pathophysiology, with bringing this program to fruition from a regulatory perspective with the FDA, and really thinking through next steps in the program. So being here as part of the community today with you all, we're really here listening to you. We're trying to understand how we can best leverage our expertise in working in the rare disease space and the programs that we've brought forward to date to understand what are your needs, what are your priorities. So we are here as active listeners to understand how we can best leverage the synergies that we have, the resources that we have to best serve the medical communities. And that's all you here sitting in the room. So again, thank you. And I'll echo Pyle's comments earlier. Please find us in the room. We have the big name tags on, and we're here to learn from you. And we welcome and thank you for embracing us. I believe there was one other slide there, which I can just comment on high level. It's okay if the slide doesn't come up. I alluded to some of our experiences to date in the rare disease space. So we are a rare disease franchise, and we've had successes from a commercialization perspective in bringing therapies to individuals with SMA. We had an approval in 2016 with our drug development program there. We continue, again, bringing it back to our commitment. So it's not just getting through that finish line from an FDA and regulatory perspective. It's what we can do next for ongoing learnings. So we are committed to continuing our development program in spinal muscular atrophy to really think through. We have an understanding that the treatment landscape changes, it evolves. The medical community continues to understand different aspects of management of individuals living with the disease. So we continue to look for how we can optimize treatment for patients with the rare disease, particularly in SMA as an example. We also had in our rare disease neuromuscular space an approval earlier this year in ALS, which ALS is my passion. Is what brought me to Biogen because of their commitment to this space in neuromuscular rare diseases. And this particular approval we had earlier this year was with an antisense oligonucleotide, so a bit different than what Pyle was talking about with a small molecule as far as what we have now with the Reata integration. For the ALS program, it's an ultra rare disease, SOD1 ALS. Only 2% of ALS patients have the SOD1 mutation. So again, it's that commitment of not shying away from what's difficult in this space and really tackling what's possible for the ultra rare disease patient population. And of course, as Pyle pointed out, with this integration of Reata into the Biogen family, we can really pull through all of our learnings from the spinal muscular atrophy development program and work that we've done, as well as ALS, and think through how we can take those learnings into what we can do to best serve you. So with that, I thank you all. And again, welcome interactions throughout the rest of the day, and look forward to future events like this to interact with you all. So thank you.
Thank you. All right, our next program, we're going to move to vatiquinone. And unfortunately, our colleague from PTC could not join us today, so I will be giving these slides. And I need to look at them. So you all know vatiquinone. Probably some of you know it by a variety of names, PTC 743, or those who have been with us for even longer, EPI 743. And this is one of those approaches that works within the mitochondria. The drug itself is an inhibitor of an enzyme, 15-lipoxygenase, and which is involved in actually protecting cells from oxidative stress. And we talked about oxidative stress within the mitochondria earlier, and this is one of the defense systems. But the activity can actually become harmful and drive some of the changes in membranes, especially within the mitochondria. And that's what vatiquinone is aimed at doing, to prevent those changes that occur and ultimately to decrease inflammation, preserve cell survival and cell function. It's been in very, very many people as it's progressed across the decades. So there are about 700 patients that have been treated with vatiquinone, many of them children. So we know a good safety record goes along with this drug. However, in the trial, the MOVE-FA clinical trial that was recently completed, the primary analysis or the primary endpoint, so in a clinical trial, we pick one measure that we say this has to change for approval of the drug. We cannot change that measure once the clinical trial has been started or once we have the data roll in. And that measure was mFARS, as you heard about earlier. And it did not meet the pre-specified or declared changes in mFARS that we anticipated seeing with the drug. It did demonstrate effects on subsets of mFARS, which Paola explained to us, and on some key secondary endpoints. And the subset particularly, and this seems to be an important one in children, is the upright stability subset of the scale. It also had an effect, an impact, a positive impact on fatigue. That being the case, that it appears to be a very safe compound, that we know there hasn't been any major adverse events that were associated with it. There were some changes in GI function, which seems to be a theme. But we did not see anything that would prevent, from a safety aspect, this drug going forward. However, because the primary endpoint was not met, there is a negotiation that has to occur with the FDA, in this case, with the regulatory agency, to see if there is, in fact, a path forward for vatiquinone. And that's what's happening right now. I think what we'll do is take questions for all of these together at the end. So I'm going to move on to Jean. Will join us to talk about Stealth project. Okay, you want to introduce yourself, please?
Good morning, I'm Gene Kelly from Stealth Biotherapeutics. Thank you for having me. Next slide. Oh, I guess I can do that, right? So just a little background on the company. Stealth has been around for about 15 years. Our primary focus is mitochondrial medicine. So as you've heard several times before, mitochondria are the powerhouse of the cell. They're essentially in every cell in the body with the exception of red blood cells. And they're most important in organs of high energy demand. So things like skeletal muscle, the eye, and the heart. So our lead compound, our investigational compound, is elimipritide. And we've looked at a number of different things from cardiomyopathies to mitochondrial myopathies. We're also looking at some common diseases like geographic atrophy or dry AMD. So 3 organs that are of interest also in Friedreich's ataxia. And again, in terms of experience with a rare disease compound, you always want to know like how well characterized is the compound? What do you know about how it works? How safe is it? You know, what can you anticipate in terms of efficacy? So we have patients that have been on the drug for up to 5 years now. And just to give you a sense of some of the other things we're looking at, right now we are planning a phase 3 trial in dry AMD. So again, looking at the back of the eye, the retina, there are regions of the retina that are very mitochondrial rich. And if you get mitochondrial dysfunction, the eye doesn't function properly. So the idea is restore the energetics and either maintain or improve function. Primary mitochondrial myopathy, this is the program that is a clinical trial that just completed enrollment. So we should have results for that next year. And Barr syndrome is a case where we've completed our trial, but we're still trying to negotiate a regulatory approval with the FDA. It's a disease that's marked by cardiomyopathy. And one of the challenges is it's such a small population that it's very difficult to get the kind of statistical power that the FDA would like to see. That a lot of times they'd like to see more controlled data. And sometimes the population is so small that it's very difficult to do additional trials. And it's another example where advocacy is just so critical in terms of educating regulatory authorities about the disease and the practicalities of doing additional trials. So, you know, we're interested in FA. We have some pipeline compounds. Elimipritide does not cross the blood-brain barrier, so it's probably not an ideal compound for some of the neurodegenerative diseases, but we do have some pipeline compounds that do cross the blood-brain barrier. And we do have one series in our 550 series. We're actually looking at preclinical work with FA. So I think the company is going to remain committed to FA for a long time. So just a little bit of background on the drug. Everyone that's gone before me has done a great job of setting up the mitochondria as a potential treatment target in Friedreich's ataxia. So, you know, just a little bit in terms of the mitochondria. When you see healthy mitochondria, it's a power grid. You see those curvature, the cristae that are depicted in the iconic versions of it, but it's that bend that allows the electron transfer. And those little orange pieces are cardiolipin. We often describe them as like the triangles in a pizza pie, make a round curve. So it's like they help hold the structure together. But when they get oxidized or, by reactive oxygen species, or they get eaten away, or you have a rare disease that doesn't allow you to make healthy cardiolipin, you tend to lose that structure. And instead of making ATP, you make lots of reactive oxygen species. Our compound seems to work by improving both the structure and function. It seems to help tether those the inner mitochondrial membrane together. And as you've heard multiple times today, we believe that mitochondrial dysfunction might play an important role in Friedreich's ataxia. So in terms of clinical trials, CHOP, Dr. Lynch, David Lynch, is doing a trial looking at the tolerability, efficacy, and safety of elamiprotide in advanced symptoms of Friedreich's ataxia, focusing on things like vision loss and cardiac disease. So this is a case where we were presented an opportunity to work with CHOP and FARA. We knew a fair amount about our compound, and they brought us a lot of expertise about the disease and helped us figure out what a trial might look like. So that is ongoing now, and it would be characterized as a phase 1, phase 2 trial. And I'll just end by thanking you and reinforcing that without your participation in clinical trials, your willingness to show up for these events and endure the hardships that come with that, and the willingness to educate regulators and government agencies on your diseases and the need for safe and effective treatments, none of this is possible. So I just want to thank you again for including us and allowing us to be here today.
Thanks, Jean. And next we have Rusty, who's going to tell us about the Larimar approach.
Thank you, Barbara. As Barbara said, my name's Rusty. It's Rusty Clayton. I'm the Chief Medical Officer for Larimar Therapeutics. And echoing my colleagues, I want to thank FARA for first hosting this event today and also for inviting us to participate. I've worked in a lot of rare diseases, and you have a wonderful advocacy organization here. They've been. There we go. We arranged that before. I'll tell you that Jen and Ron and others have been extremely supportive of us, and I'm sure of all the companies. They've been down to FDA with us. You really have a wonderful leadership and a wonderful group. So I want to thank them, and I also want to thank and echoing the last speaker, the participation of the volunteers for these trials. Without the volunteers for these trials, you do not have clinical development. And so thank you very much for participating in all trials. And if you have happened to participate in the Larimar study, I thank you. I also thank you to not let me know who you are. I'm not allowed to know that, so keep that to yourself. So Larimar Therapeutics is actually based in Ballackinwood. It's just right down the road, if 76 is considered a road, sometimes considered a parking lot. And they were Chondrial Therapeutics before then, and they are dedicated to finding a treatment for Friedreich's ataxia. Let me just go to the next. Is that me here? Yeah, okay. And I only have one slide. And the approach here is extremely simple. We've heard several times that Friedreich's ataxia is caused ultimately by a genetic abnormality that results in lower frataxin levels in people who are affected. And so the strategy here is to replace that frataxin, or I should say to supplement it. So you'll see the slide on on your left is frataxin. It has two pieces to it. One's a mitochondrial targeting sequence. It's in the name. This is how the protein gets to the mitochondria. And then you have the mature human frataxin that works its magic in the mitochondria, helping to form these iron sulfur clusters that you've heard about here. This is not actually how it looks. I don't know how it actually looks, but this is just the diagram of it. And then on the right is CTI 1601. And you'll notice there's that little red piece on the left-hand side next to the mitochondrial targeting sequence, and this is the cell-penetrating peptide. And so what happens is CTI 1601, or its name you're going to start seeing is nomlabofusp. If you thought omavaloxolone was tough, and you're allowed to just call it NOMLA for short, that's fine. It is injected under the skin, and then it makes its way into the bloodstream, and from the bloodstream, it infiltrates into the cells. And it's the cell-penetrating peptide that allows it to get into the cell. And then the mitochondrial targeting sequence takes it, once it's inside the cell, to the mitochondria. And then those two pieces, the red and green pieces, if you will, break off. And it allows the frataxin to do what it's intended to do. And very simply, this is the mechanism of action of CTI 1601. We've completed our adult phase 1 studies, and we are currently in the process of doing our dose exploration trial, phase 2 dose exploration study in adults. We will be opening soon our open label extension study, which is going to allow people who have previously participated in a CTI 1601 study to enter a longer term study of CTI 1601, and it'll be open label, which means everybody gets CTI 1601. There will not be any placebo group. So with that, I'll just again thank everybody very much for providing this little update, and we'll move forward.
Great. Put this back down a little bit. Our next speaker is from Design Therapeutics. Jay, do you want to come up?
Thank you, Barbara. My name is Jay Kim, and I am Chief Medical Officer of Design Therapeutics. And we'd like to thank FARA for inviting us to present the multiple ascending dose study of DT216 and our next steps for development for that therapeutic. We will be making some forward-looking statements. Slide always gets a few chuckles. As you know, the root cause of FA stems from the inability of cells to synthesize frataxin. And as Dr. Lynch, Dave, as Dave so eloquently reviewed before in the presentation, that there are multiple manifestations of FA that include central nervous system, peripheral nervous system, heart, pancreas, and also skeletal muscle. Now here we review the mechanism of action of DT216, our first gene-tac therapeutic small molecule that went to the clinic. On the left side, you see what happens with the disease. That is, the GAA expansion repeats cause a transcriptional roadblock. That really means that it renders it very hostile for the RNA polymerase to make RNA. On the right-hand side, we see the mechanism of DT216, which is shown in light blue there. It is a hetero-bifunctional small molecule, which means that it has two parts of the chemical. One part specifically targets the GAA expansion repeats on the genome, and the other part actually recruits the RNA making machinery in the cells. So it enables the cells to synthesize endogenous frataxin RNA. And then the intronic GAA repeats are spliced out through normal processing, and you generate endogenous mature frataxin mRNA. Now, that is the key biomarker for our drug's activity that we develop methods to measure in the clinic. Now, first, I want to go over an observational research study, which means we wanted to understand the behavior of the methods that we developed to measure mature frataxin mRNA. And we recruited healthy volunteer controls, carriers, which are people who are heterozygous for the GAA repeat expansion and are clinically normal, as well as genotype confirmed Friedreich ataxia. What you see here in the plots, healthy volunteers in gray, carriers in purple, and FA patients in blue, that most patients are generally non-overlapping with the carriers who are phenotypically normal. Now, these findings actually help interpret our clinical trial results that I'll go over in just a moment. Here, this is a high-level view of our phase 1 program, and I draw your attention to this middle column in yellow, which is the single ascending dose study that we had presented in the past, where we evaluated doses from 25 milligrams up through 600 milligrams of intravenous DT216. We did observe some cases of injection site thrombophlebitis, which were observed at the very highest doses at 400 and 600 milligrams. That helped to inform our design towards the multiple ascending dose study. And we selected 100, 200, and 300 milligrams for multiple dosing. And we selected those because that we predicted that there would be therapeutic exposures for 200 and 300 milligrams specifically, and also in anticipation that that these doses would be tolerated at the injection site. Now, some new feature in the multiple ascending dose study was that we decided to adopt muscle biopsies so that we can evaluate what happens in a disease-related tissue. And these were in the 200 and 300 milligram dose levels only because we thought it would, muscle biopsies are hard and probably best to do it at doses that we thought the drug would be doing something. So this slide summarizes drug levels of DT216, and on the right-hand side, drug levels in the blood, and it's just a plot that shows that DT216 behaves quite well, and that higher doses translated into higher levels of drug and measured in blood. On the left, we summarize here in words that the levels of drug in the muscle tissue, however, was somewhat lower than predicted in our animal studies. But as you will see in the next slide, it was still sufficient to drive substantial pharmacology or drug effects. So here we summarize the clinical activity of DT216 in skeletal muscle measured by endogenous frataxin mRNA. On the left-hand side, we see the average effects in placebo shown in gray, 200 milligrams shown in green, and 300 milligrams shown in orange. And at 2 days after the third dose, we see significant elevations in frataxin mRNA, and the average effect higher than the upper 75th percentile in FA patients that we were able to measure in the observational study. And also this effect was durable, that we saw elevated frataxin RNA out to 7 days after the third dose. On the right-hand side, we see the individual participant summary for all the patients who were in the placebo 200 and 300 mg dose levels. Overlaid are these bands in light blue, the reference values from FA patients, and a light purple, the reference ranges from healthy carriers. And it's kind of shown as you would kind of see in the children's growth charts, so to speak. Now, what's quite notable is that we had one patient here in the far right who actually achieved healthy carrier levels of frataxin mRNA. And over half of the patients treated with 200 or 300 milligrams achieved the upper quartile, upper 75th percentile of frataxin mRNA in FA patients. And I think this portends well in that we predict that higher frataxin levels would, maybe result in lesser severity FA symptoms. But this is really historic for us. We're incredibly impressed that the very first synthetic transcription factor drug that was ever developed is doing exactly what it's supposed to do. Now, just in terms of a summary of the clinical activity results, we saw significant endogenous frataxin mRNA increases in skeletal muscle with durable increases out to 7 days, that the frataxin mRNA increase had a significant dose response and exposure response. And so it just tells us that the drug behavior is rather predictable in terms of the clinical activity. Now, it is worth also saying that we did observe injection site thrombophlebitis events across the 3 dose cohorts. There were no serious adverse events, and there were no treatment-related discontinuations. All adverse events in our study were mild, mostly, and some moderate. None were severe. And the 5 AEs of injection site thrombophlebitis events, and of these, 4 were mild and 1 was moderate. So we have performed studies to indicate that the thrombophlebitis events are due to the excipient composition in the current formulation, which means it's the stuff that's mixed in with the active drug. So due to concern for potential worsening of injection site thrombophlebitis at higher doses or with multiple administration, and mind you, we're developing this therapeutic to be a lifelong therapeutic. So it has to be well tolerated for the long term, So we have shifted our focus to developing DT216 with an improved formulation to enable higher exposures and chronic intravenous administration for the treatment of FA. So like most good things, this is going to take some patience and some time, which is a tough pill to swallow for those of us who are, so impatient to bringing potentially transformative therapeutics to FA patients. But we ask you to be patient with us as we work very hard towards a new formulation, and we're making lots of progress on that front, that would be able to navigate the injection site events, and we hope to be back in the clinic later next year. But none of this would have been possible without the participation of you, the FA patient volunteers, and their caring and thoughtful caregivers, and for the support of FARA and also the scientific community. Thank you.
For a few questions. And while we're waiting for people in the audience to think about their questions, there are a few that came in online. So Larimar, it seems that the injection site reactions were common in the trial. Were symptoms associated with the injection site reactions different between people who got the drug and people who got placebo?
Very good. Can you hear me? Okay, there we go. All right, thanks. So thank you for that question. It is an injectable drug. It's injected subcutaneously, that is under the skin. So injection site reaction is something we study very carefully. In 100% of subjects who received active drug, there was an injection site reaction. In about a third of the placebo patients, they had injection site reactions. To some degree, the injection site reactions in both groups increased as the dose increased. That is, the volume injected increased. The injection site reactions tend to be redness around the site of the injection that will typically disappear within 15 minutes. Some will last a little bit longer. But that was really the big difference was the frequency, not necessarily the severity. And of course, severity, when I say that, they all were mild. There were a couple that were moderate.
That's great. So I think you answered this, but the dose, the volume of the injection increases with dose?
Yes. So
Okay.
we have only one strength. And so as we increase the dose in a multiple ascending dose study, for example, you're going to give more volume. And we know just in general, giving subcutaneous injections, as you increase volume, you're going to increase the risk of an injection site reaction in a subcutaneous administered product.
That's great. Thank you. That's very helpful. So again, for you, using data gathered from non-human primate studies, can you quantify how the drug accumulates in different regions of the body?
That was not part of our non-human primate study, so I can't really elaborate on that. In terms of the different parts, we did measure frataxin concentrations, and we did see a dose-dependent increase in frataxin concentrations in the buccal cells and the skin cells of the monkeys. But I can't speak to where it goes and how it accumulates in different parts of the body.
So the next question I think might be speculative as well if you don't have those data in the preclinical studies, but do you have a sense for what levels of frataxin increase would you expect in different tissues, specifically in the cerebellum, the dorsal root ganglia, or the skeletal muscle? So is there a difference in distribution across those tissues that you understand at this point?
So we didn't look at frataxin levels in nervous tissue or muscle or heart or kidney or liver. We looked only in the buccal cells and the skin. It was our plan clinically that we were going to go ahead and look at that. That's where the majority of the published data are in those types of cells. So
And you don't have a sense either that there's over accumulation in any particular organ.
So when you give frataxin to an organism like a monkey that does not have that has normal frataxin levels, you're going to now get supernormal frataxin levels, and that's exactly what we saw there. When in our phase 1 and our current phase 2 data, when we have given CTI 1601 to patients with Friedreich's ataxia, we're seeing increases of frataxin levels relative to their baseline, as well as relative to placebo in the buccal cells and in the skin cells. So we are seeing the increases, and the increases, it's very small numbers. So I can't say the increase is X or Y. It varies a little bit by individual.
So early in the study, early in the clinical development. Thank you for that. Okay, question for design. It seems like a key aspect to your drug being effective is its ability to maintain drug concentrations in target tissues, especially with these weekly IV injections. Do you have any data that supports the idea that the reformulation will be able to maintain high concentrations over the end of 7 days?
Wow, these folks calling in are super smart.
They're all drug developers one way or another.
Yes. Well, I can't speak anything at this time to the new formulations. I can say that we're making progress, and that they're specifically designed to be well tolerated at the injection site, but also what you are suggesting is very provocative, and I'll just say that it's very interesting and something that we would work towards. I'd say that drug-like behavior for small molecules that has well-behaved drug levels in the blood, and also go away when you stop using it, are attributes of small molecule therapeutics that that DT216 can leverage. But great question, and I'm sorry I can't answer that for you at this time, but I hope I can in the near future.
And a follow-up question, is there an open label study that will be available for those who have participated in the single ascending dose and multiple ascending dose studies?
That too is an excellent question. I think for us, for the time being, until we have a formulation that we believe that can be administered chronically, we need to address that question first before we can address questions of long-term administration. Thank you.
Questions from the audience? You did such a great job with your programs. Oh, we have one.
Ooh, I have a microphone. That's dangerous. Thank you so much for being here. I really appreciate all the work that you're doing. I'm a mom of an 11-year-old. We know two things are really critical in an FA diagnosis: number of repeats of age of onset, which makes the pediatric population incredibly, vulnerable because their bodies are developing, but also the disease is progressing quicker. So do you have a plan for pediatric studies, or any thoughts around how to make sure we have early access, because we know that early access helps with disease prevention as it moves forward? Thank you so much.
Great. Maybe our Reata Biogen team want to take that first? What's that?
First of all, thank you for the question. I'll invite my newest colleague who joined us from Reata to comment as well. We very much appreciate the question and understand the urgency around the question there. Obviously, working in the rare disease space and the SMA experience as well, we understand the milestones and the steps to achieve that. We continue to have conversations with all the relevant stakeholders to understand that next step and path forward, but appreciate the question. And being 2 weeks into this integration, obviously that is a priority, that discussions are ongoing. Kyle, anything to add?
You
Okay.
summed it up great. Yeah.
Larimar, do you want to take that?
Yeah, again, thank you very much for the question. I'm a pediatrician, and so I wouldn't actually work for a company that didn't address that need. And in fact, I worked at CHOP, where Dave works, but way back in the way back. Anyway, we have had and continue to have discussions with FDA and as well as our European regulatory agencies across the globe about when it will be appropriate to start our pediatric program. It is very much indelibly inked into our program, and we'll be starting that. The timing is still a little subject to our regulatory discussions.
Right.
All right. Well, thank you to all of our panelists, and thank you, Barbara, for a great update on our treatment pipeline and all the therapies that are currently in the pipeline and in the clinical studies. There was one pipeline candidate that we didn't discuss because it's not currently sponsored by industry. And I just want to give a shout out. There is another study that is currently open and enrolling. It is the EXRX study. It's an exercise and NAD supplement study that's being conducted at CHOP. And Drs. McCormick and Lynn, who were up here earlier, are the investigators. And they are still enrolling for that study. And it's, I believe, individuals 10 and older. 10? Okay, 10 and older. And if you are interested in learning more about the exercise and NAD study, coordinators are sitting right over there. They can answer all your questions. Another announcement I just wanted to make, or acknowledgement, I believe one of the groups joining us online today is a group that is currently at CliniLabs. So folks volunteering for the CTI 1601 study, there is a cohort currently enrolled, currently being studied and dosed. They're not allowed out of CliniLabs for 14 days, so they're joining us remotely, and I just want to thank them because it is a huge commitment participating in these studies.